So for those of you who don't know me, my name is Laura Molgaard, and I'm the Dean of the College of Veterinary Medicine. And it's my pleasure to welcome you to this ride event celebrating One Health Day, which was November 3rd, officially, and is recognized by a number of international bodies, including the World Health Organization, CDC, and others, to celebrate our fight against the diseases that affect animals, people, and the environment we share. So it's wonderful to see so many faculty and staff and students here with us today. Every November, we host a special ride event, again, in recognition of this day. And we're really happy to, for those of you who are guests to the St. Paul campus, to help share some of how the College of Veterinary Medicine is leading in this charge. So it has been a real privilege to help promote and support the college's outstanding One Health leadership, whether that's through participation in the University of Minnesota Infectious Disease Institute, UMID, or more recently, I've been asked to chair the soon-to-be-convened St. Paul campus reinvestment One Health action team. So you'll learn more about that when I know more about it. But it demonstrates the college's leadership in One Health. The depth of expertise and dedication amongst our faculty, staff, and students, and their work in research, discovery, innovation, and education powerfully advances the health of animals, people, and the environment we share, which is, of course, the college's mission. So CVM's infectious disease work flows from basic discovery to applied innovation, and our investigators explore how pathogens function, spread, and evolve. And then we translate this into real-world application, whether that be diagnostics, vaccines, therapeutics, and prevention strategies. And this continuum makes our work both deep and directly impactful. So this event today, I'm going to share a little bit about the format of how we're doing things here today. You'll be hearing a series of high-impact presentations from faculty across multiple University of Minnesota colleges, as well as seeing 2 videos from experts in the field of avian influenza, which is our topic today. The format is designed to use this limited time that we have together to showcase the incredible range of expertise and approaches that we're collectively bringing to bear on this complex challenge. So events like this help us identify new research synergies and uncover unexplored questions, as well as building relationships that lead to meaningful impact. So really happy to thank our presenters today for sharing their time and expertise, thank the audience for engaging, and invite you to stay fully engaged throughout the presentations, but also stay for the reception afterwards. There will be refreshments, and I am sorry that I will miss that part because I have to run over to the Minneapolis campus for a meeting with the provost. So ask questions, introduce yourself, and explore ways that you might work together, because that's where the momentum begins. And now with that, I'd like to get started and turn things over to Dr. Alonso Guedes, our Associate Dean for Research.
Thank you, Laura. All right. Good afternoon, everybody. Good afternoon. Oh, your mouths are full. Sorry about that. Timing is wrong, right? It's a real pleasure to be here talking to you guys. Thank you all for coming. Thanks to the presenters. Also, a big thank you to the research office folks, Tiber, Kinsey, Jessica, Shannon. Everybody worked so hard behind the scenes to get this. Special thanks as well to Dr. Carol Cardona, who helped conceive this framework. We're really excited to be here. So as Dr. Molgaard said, my name is Alonso Guedes, and I serve you as the CVM Associate Dean for Research. And I just want to talk to you a little bit about the One Health research at our college, where some of the challenges, the opportunities, as well as some future directions that we potentially could take our research enterprise towards. You guys hear me okay? Yeah? All right. Sounds good. I have 16 slides and I have 10 minutes, so that means no more than 30 seconds per slide. So please help me out here. So just last week, I was talking to the Associate Dean for Research and Faculty Affairs from CBS, the College of Biological Sciences, and our conversation was surrounding One Health, and it had something to do with, potentially coming up with some framework for One Health in the St. Paul campus. And he told me, it's like, if we call this thing One Health, about a third to one half of our faculty is going to feel completely left out of this. Okay. And so that was kind of a little bit shocking to me that we have the Biological Sciences College and a lot of their faculty would not feel connected to this idea at all, right? For me, One Health felt like very normal, very natural when I first heard of it. In vet med, we are always looking at comparative stuff. And so it's kind of like a little bit kind of part of our DNA. When we talk to sometimes with our human counterparts, the way that they think about One Health can be very different. A lot of times, it's a human-centric type of view, and they're thinking about the different disciplines coming together to take care of the human. And that makes sense, I think, from their standpoint. But the One Health thing is actually much bigger than that. And so I look at the definition, this very nice publication from the Lancet, just came out now in 2025. So it is an interdisciplinary, transdisciplinary, and multi-sectorial approach to addressing our problems. And so this is one of the first times that I hear the word transdisciplinary, right? I always hear like interdisciplinary thing. So really this is like transcend boundaries of different disciplines as well as different sectors to create solutions for lots of different problems, right? And so this One Health idea then is centered in this interdisciplinary integration. And the idea there is that this transdisciplinary transcendence of this disciplinary boundaries. So we really need to think about outside of the area that we're working with. And we want to integrate that expertise that goes beyond academia, right? We're all academicians here. Most of us are. We need to think about also outside of academia, as well as outside of what we call science. And so really like there is a lot of like, you can think of it like lots of different concentric circles that is encapsulating all of this. And it's important that we are kind of have our mindset on that particular framework, right? Now, One Health, I came to be aware of it mostly like in 2015 when I returned to the University of Minnesota and there was quite a bit of work being done. But this idea has been going on for a long, long time, for thousands of years, right? So communities, indigenous or native communities, they tend to embrace a lot of the concepts that we call One Health. Perhaps they do a very good job at that. Also, it has been embedded in religious beliefs as well as praxis and has also permeated science from the very beginning of what we call more modern science. So it's been there all along, but it wasn't until like the late 90s and specifically 2004 where this like idea of One Health was really crystallized by that commission, the Manhattan Principles of One Health that was made by the International Wildlife Conservation Society and some other people there too. Soon after that, in our college from 2009 to 2019, there were two major grants here, the USAID on One Health. And so those are very large projects. And so we have had a presence there that's very, very important. Obviously, we know what has happened to USAID, and so it's kind of perhaps a sad moment for that, but we have been on the forefront of One Health for a long time. So this One Health thing is really this sort of like a dynamically integrated ecosystem, right? And so you have integration there at all levels. Let's see if this little guy here works, or if I can make it work. So we have integration at all levels here. So for example, the shared ecosystems right here in terms of surveillance and interventions, as well as food systems over here. And in each one of them, we're thinking about different things. So we're thinking about antimicrobial resistance. We're thinking about infectious disease. We're also thinking about non-communicable diseases. And so all these things inside of each of these little circles, you'll be thinking about how those things are interconnected. And then you might be thinking about how those areas interconnect with each other and then how they interconnect with other things like economics, for example, knowledge generation, as well as dissemination, governance. And so this is like the ecosystem that we should be thinking about when we're thinking about One Health. You might be, perhaps working in a very specific circle here. Perhaps you are working on surveillance, or perhaps you are looking at disease intervention. But remember that if you have opportunities to interconnect with other systems, like, for example, food security, for example, or if you have opportunities to interact with governance or economics. So those are all areas that can be expanding towards in order to make your work a little bit more aligned with One Health and a little bit more integrated in the ecosystem. So you're not working alone, isolated. The challenge that we have is that we're organized very differently, right? So we can look at our campus and we actually have a silo, literally a physical silo in our campus, but there's a lot of metaphoric silos as well that we have. And so this disciplinary silos creates a lot of problems, communication barriers, right? So if I talk about One Health to a human doctor, for example, they might think about the human. If I talk to Dr. Cardona, she might be thinking about, viruses and birds. If I talk to Declan, he only thinks about viruses, right, Declan? That's how you're like. But like everybody uses this kind of terminology, but it's on their own concept based on what they do, right? And so this creates problems for us. Institutional and policy barriers as well is another one, right? So we have, we are organized in a certain way that's kind of becomes challenging for us to cross those boundaries, right? Data integration and sharing, extremely hard as well. If you're producing your stuff in your lab, you already have perhaps a difficult time connecting with people in your own area, in your own field. Now imagine connecting with people outside of that, right? It's another big challenge. And then funding constraints. There is really no funding out there that I'm aware of that funds One Health research, right? There is like components of that gets funded, but it's up to us to stitch those things together. And so that's another challenge that we have there, as well as internal funding, right? So our economic model inside the university is one that kind of also, pushes us towards a direction that's not necessarily One Health. And so we are organized more like in little boxes, right? As opposed to that circle, right? Where things are interconnected. We're all working inside our own little boxes, and that creates barriers. It's up to us to, once we have that awareness, to start, opening those boxes, connecting with the people around you, and then beyond that. There are, several examples within our college and university that kind of shows a little bit of the work that we're doing on One Health. And infectious disease is an area where One Health was really like expanded and it was conceived. And so that's an area that I'd say our college is very robust in terms of work in the One Health space. Here I just put one example of antimicrobial resistance and stewardship. And it's just, again, it's one small example, but that's in the infectious disease ecosystem that's really important. And it connects with, the human health part of it. It connects with the environment. It connects with food production. So this is one example that all of that's interconnected. It connects with economics, governance, policy, all of that. We also have now a very good example of interprofessional partnership. That's another area that's super important, right? Where we start then talking to each other and creating a little bit of a common language, for example, as well as delivering things on more of a One Health concept. So, for example, we have this integrated care model, which is a model where the human friends are caring for the humans, and we are coming together under the same building to care for their pets. So the entire family is cared for. There is also social work related with that. There is then opportunities for all of those disciplines to be talking to each other and think about the economics. Just yesterday, we were talking about talking with the director for a new spectrum of care clinic, and he was saying that one of the biggest challenges for people to access veterinary care to their pets, people of lower economical capacities, is transportation. Okay, it's hard for them to get around. It's hard for them to go from where they live to the clinic to bring their pets. And so all of these things are integrated, right? So we start thinking, working together. You start seeing what are the issues, what are the barriers, and then start addressing them. Because then the solutions for one problem becomes a solution for potentially other problems too, right? And we have lots of other examples here in our college, I'd say, that do a very good job in terms of working within the framework of One Health. They may not necessarily see themselves as One Health because, again, the infectious disease, which is where One Health kind of like really got galvanized, still have a very big cloud. And so people that are working on non-communicable disease a lot of times don't think as much about One Health. But we do have in our college quite a bit of that framework in lots of different areas, as you can see listed here, where we're working on problems, creating solutions for potentially a problem in vet med, but that can also be extended to human medicine. And potentially those things might be related to things like food systems, for example, which then can also be informed, right? And so we are working on some of those small circles there, and sometimes without actually knowing or being aware that we are providing that work. The other thing that I think we are doing is that it's one of the reasons why we're here today is the RIDE seminar series. And so this is RIDE stands for Research, Innovation, Discovery, and Education. It's a seminar series where the mission really is to be a catalyst for collaboration, intellectual growth, and scientific advancement. And so we want to help build this vibrant community of researchers. And so that's one of the reasons that we're doing this, One Health, that we're having these talks, because we want this to be a moment where we can connect. We want this to be the interstitial space, right? Where those concepts of One Health, of connection or connecting with each other can happen. And the RIDE seminar series is one area where we try to promote that, where we come together under the same roof and talk about things. And hopefully we go out and multiply that. Now, I said that I'm not aware of any funding that funds One Health, but there is perhaps some hope in the horizon. We have this National One Health framework that's supposed to go from this year until 2029. And really, it's a framework that will connect different federal agencies. So CDC, USDA, and Department of Interior have this sort of like agreement in place, or this framework where they should be working together whenever possible to address things in a One Health approach. There is no funding that I'm aware that's attached to that, but hopefully these agencies will start working and also provide some funding for the work that should be done in those areas. But that's an encouraging thing. When we look at our university, we just had the other day the strategic roadmap that was laid out. And so the Forward to Our Future roadmap, there is 5 imperatives there. And one of them has to do with the research and discovery. And so one of them is to advance our excellence to lead in the discovery and application of sustainable solutions to improve environmental, human, plant, and animal health. And so this is also in our university level. And within our college, we're also thinking about education. So this is still in preparation, but we are looking to creating a minor on One Health. And so this is just to give you a glimpse of things that are happening at the national level, at the university level, as well as our college, in looking towards the future. And so the idea here, I hope I was able to convey to you that's this interconnectedness, right? That we gotta be connected to each other in ways that perhaps we don't necessarily think about. Okay, there might be opportunities sitting just outside of your circle that if you're able to just reach out, that's gonna really elevate what you're already doing. And so, thank you for listening. Think about the interconnectedness. Think about the circles. Think about reading that paper on the Lancet. It's a lot of pages, but it's a very good. I learned a lot just from reading that. And so I encourage you to do that as well. Thank you guys so much. All right. And now we have Dr. Shangley in that will be talking about integrating host diversity, movement, and landscape dynamics and model of avian influenza. And I believe this is one of our videos, right? So we have some individuals that could not be here, you guys. So we have recorded.
All right. Hi, everyone. This is Shanglai, a postdoc from University of Oklahoma. I'm very happy to share my work here for the One Health Day. I hope my work can be, somehow helpful to your work. And if you want to talk more about avian influenza and bird migration, please feel free to email me. Before I begin, I want to thank my collaborators because none of the work will be possible without the support. Particularly, I want to thank Dr. Jean-Michel, who is the current PI of my research group, He's very supportive to my research interest, and also my PhD supervisor, Dr. Fred Boer from Wageningen University in the Netherlands. He helped me to set my research foundation. For highly pathogenic avian influenza, it's a well-known zoonotic disease. It has been a long-lasting worry for people. I remember when I watched this TED talk given by Bill Gates back in 2014, he woke up the stage with this barrel, saying that when he was a kid, that every family in the United States used to have this barrel full of food and water for nuclear disaster preparation. Then he started talking about zoonotic disease such as HPAI. And he said nowadays, the zoonotic disease, like avian influenza, could be more dangerous than a nuclear disaster, and we are not ready for it. Over the years, we didn't see the HPAI cause any global pandemic, but we do see the sporadic human infections over the globe. And since the second introduction into United States in 2021-22, the United States has become the country that has most human cases since in 2024, which is a big surprise to a lot of people. Human infection, human HPI is originally from wild birds. A conceptual model built for the global circulation is that the migratory waterfowl can carry the virus when they are migrating, as shown in this diagram. And this figure is a phylogeographic model result showing the process that HPAI originated from East Asia and was introduced to Europe over the years, and of course, is likely a result of waterfowl migration. And over the past years, HPAI has become more frequent and include more species than before. This figure shows global outbreak of HPAI H5 subtype, which is dominant subtype in the HPAI family. And we can see here that over the years, the number of outbreaks and also the number of species and the taxonomic family that influenced by the HPAI is increasing, and also the geographic regions are all that impacted by the HPAI also increase a lot. So this is becoming a big threat to wild animals, but not only wild animals like waterfowls, but the virus also infecting, the species that were considered low risk and also spill over to land mammals, sea mammal, and of course humans. So in my talk, I have 4 sections. The first section is about to understand the associations between host trait diversity and HPAI outbreak. Following on the trend of HPAI infecting more species, we'd like to know which species is the host and which species can host more subtypes. This diagram showing how species' functional traits can influence pathogen transmission by influencing their exposure rate, susceptibility, and suitability. And the functional traits here are the life history trait, foraging traits, and behavior traits. So with this diagram, we can theoretically associate the traits and species infection status as well as their host status. And this is made for, generally made for the zoonotic or infectious disease. And for avian influenza specifically, we listed out the waterbird species in the Europe and dug into public database to summarize whether a species has ever been confirmed with avian influenza infection and how many different subtypes it has been confirmed with. This phylogenetic tree shows how the data looks like, right? The red nose means the non-host species, and the blue nose means the host species. And we can see that non-host and host are clustered separately across the phylogenetic tree. And we find that waterbird and shorebirds are the 2 families that can host most of the avian influenza subtypes. And with that database, and also have their functional traits in the data, we did regression models to analyze the associations between the traits and the host status. And we find that significant association between traits and host status and subtype diversity. And we find that fast-living species are more likely to be host or hosting more subtypes, probably because the fast-living species would invest more energy into their reproduction instead of immunity. And meanwhile, we also find species with greater dispersal capacity, longevity, and body mass are also more likely to be a host or hosting more subtypes, probably because the dispersal capacity and longevity can increase the exposure risk, and a higher body mass can increase their tolerance to the infection, so they can infect more. A second part of this diagram is about how functional traits can be scaled up to community level and influence disease risk in the host community. The functional traits can be at the community level, can be measured as two metrics. The first one is functional divergence, and the other one is community weighted mean. Divergence means how similar or dissimilar the coexisting species are, and CWM means the trait value across coexisting species weighted by their abundance. With that framework in mind, we tested whether the functional structure of the waterbird community can explain or predict highly pathogenic avian influenza outbreak. So we used the waterbird wintering survey data across Europe from 2015 to 17, and we have more than 7,000 survey sites. For each of the sites, we have the species data about species richness and abundance, and we also filtered the HPAI outbreak data in two epidemic waves, the 2016 to 17 and 21 to 22. And with the survey side and outbreak side, we did a cross-selection process to match them. So now we know for each of the survey side whether they have an outbreak or not. And also for each of the survey set, we calculate the functional divergence and the community weighted means of different functional traits. We also extracted environmental variables, and then we use the data set to feed the GI models for HPAI. And we find that functional divergence are negatively associated with disease risk, meaning the community with more dissimilar species would have lower outbreak risk, because if these species are dissimilar to each other, they will probably have low contact probability and low transmission to risk to each other. Also, we did a various practitioner analysis and find out models using functional divergence variables can have a comparable predictive power to the model with environmental variables, especially for the epidemic wave in 2021-22. This suggests that the functional divergence in waterbird community can explain or predict HPAI well. After testing the effects of functional divergence, we decided to test the effects of phylogenetic divergence on HPAI. So we calculated the community's phylogenetic divergence matrix for epidemic wave in waterbirds and poultry in 2016-17 and 20 to 21. That's the subtype of H5 and 8. And we also calculated that for epidemic wave in wild birds only in 2021-22, and that's the subtype of H5 and 1. And we use the same method as we did for the function divergence, and we find that the phylogenetic divergence is also negatively associated with HPAI in wild birds in different epidemic waves, meaning that the impacts of community structure can predict the disease risk across waves of different subtypes. Right? However, we didn't see the same pattern in poultry, meaning the mechanism of HPAI outbreak in poultry is different from wild birds. But we find that poultry outbreak is positively associated with abundance of breed species wild region, suggesting that the breed species is the key for outbreak in poultry rather than wild bird community structure. The second part of my talk is about how waterfall migration influenced HPAI or avian influenza transmission. A host migration is associated with pathogen spreading. It's been documented in different host and pathogen systems. And for example, avian influenza in the birds, Ebola in bats, Lyme disease in birds as well, and West Nile virus in birds as well. And my former colleague Claire summarized these associations into different types. Generally speaking, that migration can increase or decrease prevalence. And I would like to particularly point out the migration escape because you will see this from my modeling results in the following slides. And migration escape means the migratory birds will leave the habitat where they have a high infection risk due to the accumulation of pathogens in the environment. So migration allows them to lower the exposure risk and lower the prevalence and disease transmission in the population. And this is the first study I did to understand how waterfall migration would influence avian influenza transmission, and it was back in 2014 and 15. At that time, we're still trying to understand whether a non-duck species like migratory goose, whether they can disperse the pathogen from their breeding ground down to their wintering ground in Europe. So I received more than 9,000 swab samples from collaborators, and this data were collected between 2002 and 2013 from 3 geese species along the migration route in Europe, and we fitted a GLM model with model selection averaging to understand the infection dynamic in the population. And what we find is that the geese were free of infection during the breeding and spring migration, and the prevalence in the population increased over the wintering period across all 3 species, and suggests that they only pick up the virus after arriving at the wintering ground and develop their infection in their populations. That means the migratory goose are not long-distance carrier for avian influenza, but rather get exposed locally and amplify local infection after they arrive at the wintering ground. But I need to clarify that this result is based on the data collected before 2013, which may not reflect the transmission of current viruses. And from a theoretical perspective, we were also, and we still are, curious about how migration strategy affect infection dynamics. We designed migration pattern of habitat used based on 2 strategies. The first one is the number of sites used during the migration. We call them, sequentially, the site is sequentially used. We call them serial site used. And the other one is whether the population spread over the landscape and using multiple sites at the same time. So we call this a parallel site use. And we build different combinations between these 2 strategies. Meanwhile, we designed migration synchrony to follow a normal distribution inspired by this observation of wintering birds dynamic. And we find we have different step lengths to model their migration synchrony. A shorter migration step length means most of the individuals prefer to leave a habitat at the same time. And we also have this ISIR model with the environment transmission, and we combine them all together into a theoretical modeling framework. And here's the result. We find that a serial strategy can reduce the infection prevalence in the population because the individuals can leave the site with cumulative high infection risk to settle with a low risk. This is supporting the idea of migration escape. Well, when the population using multiple sites at the same time, that will advance the transmission development because when the population split into multiple sites, and they will encounter larger infection risk from the environment. in the scenario of a combination of serial size and migration synchrony, we find that when migration is very synchronized, which means more individuals migrate at the same time, the infection will be lower compared to large migration stablings. That's because if all the individuals move together, there will fewer individuals left behind to pick up the infection in the accumulated in the environment, right? So most of the individuals that already left will be safe. So this tells us there might be optimal migration strategy for the bird to reduce the infection risk during the migration. A third part is about waterfowl and habitat availability. The background of this study is that landscape change, particularly in East Asia, which is one of the major migration corridors for waterfowl and hotspots for HPAI outbreak. And this figure shows the loss and the gains of wetland, surface water, and rice paddy in East Asia, and especially in China. And rice paddy is very, very good food source for waterfowls. So we'd like to know how this landscape change influence waterfowl distribution along their flyway. So in this study, we collected eBird records of 8 waterfowl species along East Asia, Australian, Asian flyway. We also collected a habitat data layer between 1992 to 2012, and that includes wetland, grassland, and cropland area. And we fitted GLMs to identify all suitable sites along the flyway. And as you can see, that each species have a species-specific distribution range along the flyway. For example, gray-lag goose. They have a relatively short but pretty wide distribution range in the flyway, while greater white-fronted goose have a long but relatively narrow distribution range, right? And for each of the species and their habitats, we also calculate the habitat availability matrix, like, for example, their fragmentation and isolation matrix. In the left figure, the red pixels means habitat loss and gray pixels means habitat increase. We see a lot of heterogeneity across the habitat types, like water, grassland, wetland, and wetland fragmentation, etc. We see a lot of heterogeneity here. If we plot wetland availability change ratio across latitude, we can see a much clearer pattern as illustrated in this panel. And the wetland in south part of the flyway, low latitude area, such as China, Yangtze River, and Japan, that's the area with the most severe wetland loss. Well, for the wetland in north area, which is the high latitude, that wetlands are improved a lot. Right, basically, along the flyway, in the south part, there's a lot of habitat wetland loss, but in north part, there's a lot of wetland increase. But not all the species were equally affected by that. For the species with long and narrow migration corridor, such as great white-fronted goose, they will probably face much more stress from the wetland loss, because if they lost their habitat or wintering habitat in Yangtze River in Japan, and then they have nowhere to go to, right? However, for other species like gray-lag goose, they have a short but wide distribution. So even they lost some size in their wintering ground in China, Yangtze River, but still they still have some flexibility to go to other closed habitat as a alternative, right? So this says the species will have a species-specific response to the habitat change. The last section of my talk is I'd like to talk about how this landscape change influence disease dynamics. As a reminder for, we just talked about landscape change in Asia. Um, Following that up, we'd like to see how that would further influence disease transmission driven by waterfall movement change. And to answer that question, we integrated three components: the host movement, and the virus transmission, and the environment change, which is the habitat change in here. So we integrated three components to build agent-based model to do simulations. And this agent-based model we have just published a version 2. In the first version, we use remote sensing data of grassland, cropland, and also the whiteland availability from 1992 to 2012, and eBird data to feed the GI model to predict or identify suitable habitat for greater virus frontier goose. And for each of the sites, we calculate the percentage of habitat area loss. And then we cumulatively remove sites based on the area loss from low to high. And we totally have 6 scenarios of habitat loss. And then we generate networks by connecting the remaining sites if the distance is shorter than the migration step length. So we have 6 migration network scenarios. After building the networks, we define a series of action or migration rules for 10,000 agents or birds in our agent-based model. And generally speaking, we use this animal movement model called Migration Flow Network, published by Case Taylor back in 2016. And the movement were decided by the habitat quality and the distance between the sites. And generally speaking, the agents or the birds will compare the potential size they can go to in each of the steps, and then choose the one that can that they can have more resources, but with a relatively short migration distance. At the same time, during the migration, the agents their movement was also regulated by the number of stop sites they have and their body condition and energy consumption and accumulation along the migration. After defining their movement rules, we integrated all the networks into our into the net logo, which is a platform we use to simulate our agent base. We also include the ISR model to simulate pathogen transmission. Here is the results. We show the results show that when we keep removing sites from the network, the winter birds have to relocate northwards due to the severe habitat loss in the Yangtze River basin. This will significantly short their migration distance, and that lead to an earlier and larger birth aggregation in the remaining sites. And this slide shows the results of infection dynamic among agents. First of all, we see that infection prevalence can quickly reduce due to migration and form another minor peak when the birds aggregate at the winter site after migration. This aligns with the migration escape concept. However, when we keep removing sites from network, especially in the most severe habitat loss scenario illustrated by the right line here, the migration escape effect was reduced. Right? That's because the agents finished their migration early and form a larger aggregation with more susceptible individuals in the population. So we can see the second peak appears earlier and form a larger prevalence. Meanwhile, our simulation also shows that with a more severe habitat loss, there is a higher risk of avian influenza outbreak because the sites are better connected due to the intensive use by the agents in our model. Then we want to further include more remote sensing data with higher resolution and using telemetry tracking data to calibrate our agent's movement in the model. So we did this model version 2. And this time we included 79 telemetry trackers that collect data over 2 years. And we also included more geospatial data that describe the quality of the habitat. And then we extended transmission model. Now we have transmission in wild birds, and also we have transmission in poultry. And when the wild birds encounter poultry, we have spillover events from wild bird poultry, and we calculate co-infection in the poultry and the virus reassortment in the poultry as well. Here's the result. From left to right, there are these are the locations telemetry tracking data, and based on that, we use dynamic Brownian bridge movement model to identify the habitat used by the birds we tracked. And with that side and eBird side environment variables, we feed the GIM model to identify suitable habitat between 2000 and 2015. And the birds have, we can see here, the birds have more suitable size in the north part of the flyway, but they have less in the south. Just like what we find in the previous study, right? The waterfowl losing habitat in the south part of their flyway and have more habitat in the north. And then we use these networks put these networks into our agent-based model, do simulations. And we generated this distribution pattern between the scenarios, and we did piecewise structure equation model to analyze birth distribution and the drivers in these two scenarios. And we find that in 2015, the environmental variables such as rice paddy, wetland, and surface water becoming more important in determining birth distribution, suggesting that landscape change over the years have a significant impact on the distribution of water birds. Here's the results of infection dynamics. We see that in scenario of 2015, illustrated by right line, the infection prevalence is always higher, and the most important one is the co-infection cumulative reassortment in poultry is substantially higher in 2015. Since the reassortment is associated with viral diversification and the emergence of novel subtype of avian influenza, this result suggests that landscape change, which is the only different variable between the scenarios, landscape change can increase the potential of subtype emergence and viral diversification. And this figure gives us a little bit more detail about how landscape change increased the co-infection and reassortment in the poultry population. We took two locations as example. The first one is the habitat, a stopover habitat in northeast China. Here we see over the years from 2000 to 2015, there are more wetland and rice paddy available for the birds in our model, which made this site more attractive to the agents in our model. So we have more agents visiting this site, and they bring more virus and share more virus to the environment, and we can expect more co-infection and reassortment in poultry population. And the other example is Poyang Lake, which is one of the most important wintering sites for birds. Although in Yangtze River Basin, the birds are losing their wintering site, but some sites are expanding, like Poyang Lake. So over the years, there are more wetland area available for the birds, which also made this site more attractive for the birds. So more birds coming here and they share more virus, and we can expect more co-infection and reassortment. So this gives us a more detailed vision about how landscape change can influence the infection dynamic by influencing water bird movement. A summary to my talk here today. There are a couple of key information I'd like to share. First one is host functional traits can predict which species are host and can carry more virus. And the second is greater functional and phylogenetic divergence of host community can reduce disease risk. And starboard side user migration synchrony can influence infection dynamics, and matrices amplify local infections. And species with narrow distribution range are more vulnerable to habitat loss in East Asia, Australia, Asian flyway. And last but not least, conserving habitat can reduce the infection, disease infection, and viral transmission spillover and emergence of novel subtypes. Thank you for your attention. It's a great pleasure to present my work here. Please feel free to reach out to me for any questions if you have. Thank you so much.
All right. What a presentation. And just to remind you guys, this Dr. Ian is a postdoc still. So imagine, 10 years from now. So yeah, amazing stuff and amazing integration, right? We were talking about earlier. I saw things that I use in anesthesiology right there, some formulas, medicine, electroengineering, really, really amazing stuff. All right, so next up is our own Dr. Dana Franzen-Klein. She had a little bit of a family thing to deal with, so she couldn't be here personally, but she recorded a short video that we're going to play for you guys. So our Raptor Center folks working on One Health.
Hello, everyone. I'm sorry I couldn't be there in person today, but thank you for the opportunity to talk about the Raptor Center's work evaluating highly pathogenic avian influenza H5N1 disease epidemiology in North American raptors. My name is Dr. Dana Franzen-Klein. I'm a clinical wildlife veterinarian, the medical director at the Raptor Center, and an instruction in the veterinary population medicine department. In March of 2022, we were faced with an emergency response situation. Highly pathogenic avian influenza had arrived in the state, and we needed to act quickly to ensure that we could maintain our daily operations in the face of this highly contagious infectious disease. We set up and refined extensive biosecurity and quarantine procedures so that we could continue to admit wildlife patients, including those actively infected with HPAI, and prevent disease transmission within our facility. We also wanted to contribute to the bigger picture of what was happening in our native wildlife during this outbreak by conducting disease surveillance for our region and advancing knowledge of the disease in raptors by characterizing the clinical presentation and conducting a serology study. For disease surveillance, we have been screening every wild raptor for active infection with HPAI via polymerase chain reaction or PCR testing at the time of admission to our hospital. Since March of 2022, we have tested over 4,663 individual raptors for HPAI. As of October 20th, 225 of those birds have been confirmed positive for the circulating H5N1 strain of HPAI, and 39 individuals have preliminarily tested positive, with final confirmation pending at the National Veterinary Services Laboratory. Our disease surveillance data is publicly available on our website, and it's updated routinely as we receive results on new admissions. This data provides information on disease prevalence in our region and contributes information about how the disease is behaving in wildlife. For example, our raptor samples have documented the arrival of new viral genotypes to the region and have been some of the first positive detections marking the arrival of the disease during fall migration. Along with disease surveillance, we have also extensively documented the clinical presentation of this HPAI strain in wild raptors and characterized the disease pathology with postmortem examinations. Most of the raptors that were admitted to our hospital because they were infected with HPAI were either deceased or showing severe clinical signs at the time of arrival. The most common clinical presentation is severe neurologic signs, such as seizures, loss of balance, and altered mentation. However, we occasionally admitted a bird with very subtle clinical signs, which was an important lesson to learn and share with the broader wildlife rehabilitation community. To date, we have admitted 2 birds that were actively infected with HPAI and survived the infection, 1 great horned owl and 1 bald eagle. Prior to this current outbreak, it was thought that HPAI was almost always, if not always, fatal in raptors. However, our serology study has proven this to be incorrect. Along with testing every wild raptor for active infection at the time of admission, we are also collecting blood to look for evidence of past infection through antibody testing. This serology work is still ongoing, but the preliminary results have been quite astonishing. From September of 2022 through April of 2023, we tested 264 raptors for antibodies to HPAI. 60 individuals representing 6 species were antibody positive, indicating that they had previously been infected with the circulating strain of H5N1 and survived that infection. We saw an exceptionally high prevalence of antibodies in bald eagles, with 53% of the birds tested having antibodies to H5N1. While this ongoing outbreak of high path AI has been exceptionally challenging for wildlife rehabilitators, it has provided an opportunity to learn more about this disease in North American raptors. The information we have gained through this research at the Raptor Center has not only improved the clinical care of these birds in rehab, it has also provided insight into how this disease may impact raptor populations as a whole. Thank you very much for having me.
Very good, very good. How are y'all doing? You okay? All right. So we have a little bit more time still. So next up is our own Declan. Declan's here is going to talk about surveillance and H5 in wildlife. Here you go, my friend.
Thank you. Right. You're all thinking, what am I doing here? I mean, I work at honeybees, right? And actually, that's a common mistake, because I work on all things viruses, as Dr. Alonso has mentioned. Give me a virus and I will try and understand what's going on. So to those who are anyone working on honeybees, don't worry, I'm still working on honeybees. Dairy, I'm still working on bovine leukemia virus and IPAP. And for the swine group, yes, I'm still working on swine influenza and as well as PRRS. So what am I doing? So my background is virology. I'm very much interested in how viruses evolve, the ecology, where they exist, how they change. And the overriding conclusion I always reach is that we know absolutely nothing about viruses, because they keep changing. They keep doing different things. And so when H5, we're talking about H5 here because we actually was monitoring H5 initially, but we're doing more on that. But the H5N1 outbreak is something really different to what has happened previously. It's a virus that just continually persists, and it hasn't gone away. You usually get succession and rapid succession, but this H5N1 has been around for a bit. So how did they get me to move away from honeybees and from swine and from dairy into wildlife? And it's because you're being in the right place at the right time. Dr. Arno Wunschmann there, you can see sitting in the audience. We had a few discussions, and he was saying at the time, when we were getting all the wildlife coming in, many different species were starting to show symptoms, and he could positively identify them as H5N1. He said, there's something else going on here. Would you be interested in looking at it? And because I use techniques that are common to general virology, we end up developing a pipeline where we can collect samples, we can process them, and get data in a real timely manner. But to do this, you need samples. And so with that in mind, Arno got the VDL involved. He then made few phone calls, and through the LCSMR and the funding, then the Natural Environment Research Trust Fund, we were supported as of this year to create a network. And that's one of the big pluses for this project. It's creating a network where we can get samples sent to us for analysis. And this network includes, you saw Dana produce from the Raptor Center. The Raptor Center will send us samples. We have the Wild Rice, or the Rehab Center. They will send us samples. We are working with our tribal nation partners. If they see anything peculiar, they'll send us samples. And so it's quite an ambitious, dynamic project. I mean, you can see even listed there is the zoos, Minnesota Zoo, etc. So we have quite a dynamic group of people, and that's not easy. That's not trivial. It's getting them to come together and send us samples. The budget is to look at 9,000 samples. So we're seeking to process 9,000 samples, and we've targeted that we would go to a 50-50 split. So we'll try and look at birds and mammals. So it's not just water birds. Within the bird group, we're trying to look at a small percentage of water birds, and then any other birds that we can see that comes through these facilities that it's worth sampling. The other partnership in here is Dr. Tiffany Wolf. You can see her and her team. The role that Tiffany plays with the team is to get the data processed in a way that it's available online and you can monitor it. And so in the screen here, there's a website that we have posted through the diagnostic lab. If you go through the diagnostic lab, you'll be able to access this data set. It's a dashboard that updates weekly as to sort of the effort we've put in. The effort is at the moment, as of Monday this week, we've sampled 2,428 samples. Okay, these samples have been processed. They've been screened for influenza, a general influenza screen. Out of those positive, you can see there's 41, and those 41 have been processed again to see whether there's H5 signal, and they've all been H5 positive. What's also interesting is that you'll see that the data is currently split into what happened over December, the winter months, the summer, and now now we're going to fall, and you'll notice the two peaks. So as you just heard this from the first talk about the migratory pattern, there's obviously a good tie in terms of migration, birds assembling, and the spread. What we've also done is created a map, a distribution map, and this was very important for the funders, is to say, we just done a focusing on one area within Minnesota. We actually even getting samples on Wisconsin and possibly in Iowa. But the principle is that when we look at this map, I'm sorry, I should have had a bigger picture. You can actually get a spatial idea of where we're finding positives for influenza. This is early on. As I said, we just started this year. We are hoping to get at least a 2-year data set to try and understand where we're going. Now, my lab gets involved a bit further down the line, where we start saying, right, we know the positives, so the positive tests are done for the VDL. When we have positives, the samples get sent to my lab, and then we do some additional work. We'll do some whole genome sequencing, and we'll also go, and once it gets confirmed by the national lab that it is H5N1 and a high path variant, we will then take it to our BSL-3 facility and then grow the virus and try and find the virus. Okay. As of today, we have 3 isolates from peregrine falcons, and 2 from bald eagle. So even my partners don't know this. So Arno, you didn't know this. So we've grown, as of this week, we got 5 positives. What's interesting is one of those samples we were able to get all the way through whole genome sequencing, and there's a tree on the right here where you can see where our sequence is sitting. That fits perfectly with the classic H5N1 group, the 2.3.44b. And in fact, we've done enough phylogeny to say this is a D1.1. And this was done in 1 week, right? So this is what we try and do. We're using a high throughput method to go from screening, sequencing, and have an isolate ready for future work. All these samples are also frozen away for any further sort of screening for surveillance of what other species we can find and other pathogens that we have in the wildlife resource. And I'll end off by saying, and that's the principle behind this, is creating this network so that in the future we have a network of partners that if anything happens, that this is now H5N1, but in the future if any other pathogen comes into play, we will hopefully engage the same group, the same community, and switch to the next pathogen that might be threatening us.
This is an amazing example of how things come together in the space between us, right? Where Arno and Declan met somehow in the hallways and had a conversation, and here we are. And so this is the one example of this interconnectedness. We had programming changing at the moment here. We will play the video from our NIH partner at the end. We are going to have now Beth Thailand, is that right? Thielen, thank you, from our medical school that's going to talk to us.
And do I need to do anything to advance? Okay. Good afternoon, everyone. Thanks for having me. So I am a physician scientist by background. So I am trained as both an internist and a pediatrician, and I practice as an infectious disease subspecialist in our Masonic Children's Hospital on West Bank of our university. So I'll bring the perspective of working closely with public health, which is something I have done since I was a fellow in infectious diseases. I've had a partnership with our infectious disease diagnostic lab at MDH in downtown St. Paul, and working with our epidemiologists there. And I think just to riff a little bit on the theme of like networks, this network that I'm involved in is this Pathogen Genomics Center of Excellence. And this was really founded coming out of COVID and our experiences with things that went well and things that didn't go as well. And this is an initiative funded by CDC. And sorry, I think there was supposed to be a little logo up there, but there are 5 sites that were selected around the country. Minnesota, basically Boston, Harvard centers, Virginia, Georgia near CDC, and Washington, the Pacific Northwest, where there was good, strong public health labs that could do pathogen genome sequencing. And as a requirement of this grant, they had to have an academic partner. And so Minnesota was selected as one of the sites. And I have the privilege of leading the University Academic Partnership, which involves faculty across multiple schools, including the Veterinary Diagnostic Lab. Focusing here, my research interest is in respiratory pathogens. And so I highlighted a subset of projects related to H5N1. So the first of which was when we first started, we wanted to do a tabletop exercise. We wrote a mock scenario that I helped create. I had heard about the life infections in mammals. And so I said, what if there was a hunter that got exposed to an infected animal? And my research lab it gets surveillance samples from greater Minnesota. And so what would happen if we detected a sample in our research lab? How would we communicate to the public health lab? So sort of an example of thinking through some of the cultural differences between academia and public health. And I will tell you, there's a lot of different approaches there. We also are actively involved in curriculum development for incorporating molecular epi into our training for our trainees. And so this is housed in the College of Pharmacy and the Medical Laboratory Scientist program. And they have an online course that's a lot of curriculum, and then they're going to be rolling out a hands-on lab for sequencing-based diagnostics coming soon. There's a lot of, also, it's kind of the Wild West in terms of how methods and how people analyze data. And so part of this is a lot of committee work, taking best practices from across all the sites and coming up with ideally some common ways of analyzing data and reporting them into national repositories. Particularly, we're looking at how to analyze H5N1, knowing that it's a segmented RNA genome, and thinking about how to grapple with the reassortment problem that Dr. Yin talked about earlier. We're also thinking about benchmarking on the wet lab side. So how do we use common workflows and decide what's the best way to approach some of these problems? And so the network is sending out standardized samples with RSV, influenza, and SARS-CoV-2, and we're using our best practices and then sort of comparing how we're doing things and trying to come up with some best practices. And then finally, my lab is leading a remote household study. So we're mailing out collection kits to households that enroll with us, and then they're mailing us back samples when they get sick. And so much like the map that Dr. Schroeder just showed, we have membership from various counties around Minnesota, including and then Wisconsin and Iowa, that were mailing us back samples. So hoping to solve one of the challenges of getting samples from less severely ill people and from a more distributed geographic range. So with that, I will wrap up and hand over the mic.
Thank you, Dr. Thielen. Amazing stuff from pediatrics to bears. Very good. So next up, our own Dr. Cardona here. Thank you, Carol, for helping with all of this.
Sure. I only have 5 minutes, so get out of the way. All right. Okay, give my remote. All right. So I just want to say we can't forget about the poultry, not only because it's a very important food source for an upcoming holiday, but also because if you're calling it high path AI, you're talking about poultry. It's not high path AI if it's in a wild bird or in a cow. And for those of us who talked about H5N1, never commit yourself to an N type. It's H5 influenza goose Guangdong lineage. Say that and you'll be safe. Okay, so because we already have an H5N2 here in Minnesota. So we've only been doing Zoom now for 5 years? So forgive us. So you heard a lot about H5 influenza goose Guangdong lineage. It started in 1996 among Chinese geese. And my good friend Henry Wan was the poor graduate student who had to go out and sample those geese. What he found was a few dead geese, about 40% morbidity among the group. And he also found Newcastle disease. So they thought it was Newcastle disease for quite some time. They found 2 strains of Goose Guangdong lineage viruses. The one that we're dealing with today is number 2, the isolate number 2 from that group. Henry Wan is now at University of Missouri, for anyone who wants to talk to him. So middle. There. Okay, so poultry have had significant roles in the perpetuation of this Goose Guangdong outbreak across the globe. Significantly in China and in Southeast Asia, the virus was spread through poultry trade and became endemic in that region, which has then driven this global expansion throughout the world. We also have seen significantly in Bangladesh, Egypt, those viruses continue to cause infections in those areas. And in the U.S. in 2014-15, we had a domestic poultry perpetuated outbreak. What this tells us is that although it's being perpetuated at this time by wild waterfowl and global movements of migratory birds, this can be a lot worse. So we control poultry outbreaks by stamping out, in other words, killing the susceptible host. This is caused by a virus. And so for a virus to replicate, it needs a host. If it replicates in poultry, we stamp out all of the susceptible species around it to prevent it from spreading. This is an expensive proposition. It's not sustainable for the type of outbreak we're experiencing today. From 2022 to today, we still have active outbreaks. So this is not a procedure that's going to work to end an outbreak that's perpetuated in other species like wild birds or cattle. And it's not going to clean up the environment, but it will stop those poultry from spreading the infection. And it's the only control option we have at this time. So because H5 influenza will be with us for the foreseeable future, and it is still fatal for poultry, almost 100% fatal for poultry, we need to learn to live sustainably with this infection. So the secure poultry supply plan is the most unheard of plan in this college, I think. We've been around since 2004 at the University of Minnesota. We use science to make better strategies for outbreak response and control. We take reality and use science to make it better. Okay? So, and I encourage you to visit us. So normally what we do is we help producers and regulators figure out how to recognize not infected poultry flocks. So in an outbreak, that center red dot is an infected farm. There's a circle drawn around that farm, a 10-kilometer circle that identifies the most likely to be infected farms in that area. Those most likely to be infected, then you have to figure out if they are infected or not. It's easy to tell when something is infected, but to figure out if they're not infected, you need a complex algorithm to determine that, and that's what we have done. We also have worked to detect virus early. So when the infection first happens on a farm, using existing data, how can we know that? And how can we figure out how the virus spreads from barn to barn? We can speculate that it's because of people or it's because of rodents, but how do we really know? And how could we create mitigations between barns to break up really large farms so that we don't have to stamp out all birds on a farm and we can divide up some of those really big farms, right? Really big farms today have 7 million birds. Harvest uncontaminated product from infected farms. So you may have remembered when the price of eggs went up to $6 a dozen. That was because of those really, really large farms. And on a really large egg farm, you can have a cooler that is filled with eggs that were laid before the birds became infected on that farm, and also may contain eggs from other farms. So we think we can figure out how to safely figure out which those eggs are and move them off that farm and use them for human consumption safely. And finally, we're working to develop a risk management framework for vaccination so we can identify the farms in poultry-dense areas that are best to vaccinate to reduce the potential for spread from farm to farm, designing surveillance for the detection of virus in vaccinated flocks. Remember, if you vaccinate, you're going to reduce clinical signs, and that's one of the key ways we recognize that the virus is there. So figuring that out is how we're going to do that. And finally, estimating the impact of immunity on outbreaks, because at the end of the day, even though we're researchers and we care about science and we just want to publish our results, we still have to tell the politicians how much money we saved them, right? And so finally, this is our reality, and this is the piece of this is how we are trying to save money and make this a more sustainable and more survivable outbreak.
So we are coming to an end of this session, you guys. The NIH speaker, the video is 14 minutes long. Carol, yeah.
The video is about the cattle and poultry outbreaks in Stearns County of HPAI, of H5 influenza last year.
Oh, that felt good, didn't it? Thank you, Carol, for that. So, nice video to watch, you guys.
Hello, my name is Martha Nelson. I'm at the Division of Intramural Research at the National Library of Medicine and the U.S. National Institutes of Health. It's my pleasure today to talk about a multi-species outbreak of H5N1 in cattle and poultry in Minnesota. So this is a time-scaled Bayesian MCC tree of B313 viruses in 2024. The viruses have been colored by state of collection. So you can see the virus originated in cattle in Texas in early 2024. During January and February, it diversified into 7 distinct clades. And rapid cow-to-cow spread of B313 in U.S. cattle occurred through interstate cattle movements. You can see how each of these clades followed a different route, often beginning in places like New Mexico or Kansas, and then quickly diffusing. In this tree, you can also see frequent spillover from cattle into poultry, wildlife, domestic cats, and humans. You can see some of these big multi-species outbreaks in states like California, Michigan, Colorado. Some key observations in this tree is that the entire California outbreak was sourced by a single virus introduction from Idaho. That's this clade 5 in red. Further below, in clade 7, is the Colorado outbreak, which descended from Wyoming. And then clade 6, which we're going to be talking about more today, is where most of the Minnesota viruses fall. And you can also see that long branch length leading up to that cat, that an Oregon domestic cat fatality was connected to raw pet food sourced from Minnesota turkeys. So you can extract a lot of information about the outbreak just from the tree alone. But while we've had a lot of spillover from cattle into other species, there's no strong evidence of poultry to cattle transmission. Here's a depiction of all the directions of spillover of H5N1 in North America involving all these different genotypes. I've highlighted here on the right the B313. It's gotten into, once it became established in cattle, all the different types of species that it spilled over into, including poultry. You have direct transmission from cattle to humans, as well as cattle to poultry to humans. But as I said in the prior slide, there's really not a lot of evidence of poultry to cattle. Here's another close-up of the tree for the California outbreak. And in beige, these are cattle viruses. Poultry viruses are in blue. You can see all these independent spillovers from cattle into poultry in the state of California. But none of these blue clusters have beige viruses within them, indicating that once the virus gets into poultry, it doesn't tend to come back into cattle. So Minnesota is a place where I've worked a long time studying influenza in swine with Marie Culhane and avian with Carol Cardona. And Minnesota is interesting. It's the #1 producer of turkeys in the United States. And in this central region, you also have high densities of chickens, turkeys, and dairy cattle all raised together. Here's just a more detailed map of, for example, Stearns County in the middle of the state is a place where you have high densities of cattle and chickens and turkeys. So the B313 virus was introduced during the spring of 2024 into Minnesota 3 times based on the phylogenetic tree. We infer all of those 3 introductions came from South Dakota. 2 of those introductions were clade 6, and those led to large multi-species outbreaks that farm-to-farm spread spanned multiple counties in Minnesota. 1 introduction of clade 7, also inferred to have come from South Dakota, mostly spreading cattle, but was also detected in turkey in Pipestone County. I've worked with Pratyush Samatura at University of Oxford, who's been looking at the cattle movement data. And these are some figures he's made that shows the interconnectivity of Minnesota live dairy cattle movements with Iowa and South Dakota. You can kind of see that southwest corner of Minnesota. There's lots of movements between South Dakota, Iowa, and Minnesota. So that's consistent with this idea of South Dakota being very connected with Minnesota in terms of their cattle populations. This is a figure that my postdoc Alvin Crespo-Bellido made, looking at sort of the genotypes that have been found in Minnesota since the beginning of the H5N1 outbreak in Minnesota in early 2022. You can see in wildlife and in turkeys and chickens, you had this B21 genotype was dominant, then later B13. Then you had a big outbreak of B36 towards the end of 2023 in turkeys. And then in spring of 2024, you have the arrival of B313. And you noticed you don't see that at all in wildlife. This was something that became dominant in cattle and then spilled over to poultry. And the timing of this is that it was first detected in Minnesota in April 2024, and then subsequently in chicken and cattle. This raised a question, did poultry to cattle B313 transmission occur in Minnesota since the turkey outbreaks were identified almost 2 months before the first cattle case, which wasn't detected until late May in 2024. So this is a close-up of one of those major introductions of B313 into Minnesota cattle. And what you can see here is high intermixing of viruses from cattle and poultry. You can see up at the very top of this tree, those are those early turkey outbreaks in early April 2024. And then later in June, you have these outbreaks in cattle. So if you were to just superficially look at this tree without any outside information, you would infer that this virus was first emerged in turkey in Minnesota, and then it spread through multiple introductions into cattle and also independently into chicken. And that you really don't see any spread from cattle into poultry. But one of the important thought experiments is to think about how much sequence data is missing from different host species. how much sequence data would be missing from poultry versus how much would be missing from cattle, and what kind of biases go into what kind of data is available in the inference of this tree. And through talking with experts and learning more about how surveillance is done, H5N1 is a highly pathogenic virus in poultry, and so it's picked up very quickly. It's required that samples are sent to USDA for testing. So you're going to have pretty good surveillance of H5N1 in turkeys and chickens, and you're probably going to have a sequence for all the outbreaks in Minnesota. In contrast, H5N1 was a new disease in cattle in the spring of 2024. There was a lot of uncertainty about how it would be handled if you found H5N1 on your dairy farm. And more importantly, it wasn't as lethal in cows as in poultry. Also, there was no requirement that it be reported. And so it's estimated that very high rates of underreporting happened, not only in Minnesota, but in lots of states during that early emergence of H5N1 in the United States. And potentially more than half of dairies that were infected with H5N1 during the spring of 2024 may not have reported it or even sent samples for testing. So one of the takeaways is that phylogenetic is a really useful tool in certain contexts. I mentioned, I just kind of scratched the surface of how much we can infer about the B313 outbreak at a national level. We can see that the Michigan outbreak did not seed the California outbreak, and nor was the Colorado outbreak the source of the California outbreak, even if kind of temporally it looked like these were the biggest outbreaks in the country and might have been linked. You can see, that the virus was introduced multiple times independently into Minnesota. So even if you had outbreaks in similar counties at similar times, they might have actually arrived there through different pathways. We can do this kind of detailed molecular epidemiology to figure out where cat fatalities were sourced from. And, in a place like California, where by the time it arrived there in August 2024, there was kind of more of a protocol about sampling, and there was better sampling in cattle. And so when you have good background sampling in cattle, you can get really good resolution on the frequency and direction of interspecies transmission. You can really clearly see all these independent spillovers into poultry. When you don't have good background data from cattle, when you're missing sequence data from a lot of outbreaks, you could get a really incorrect inference on the direction of interspecies transmission. And, we worked a lot on this during the COVID-19 pandemic on SARS-CoV-2, and we've developed new phylogenetic approaches to account for missing sequence data, which we've applied for a lot of things, including H5N1 in certain circumstances. But you can only do this when an outbreak at least is reported based on PCR data. So this is really valuable if you have PCR confirmation, but you don't have a genetic sequence. And then you can kind of use these simulation approaches to account for missing sequence data. If you don't even know by PCR if an outbreak occurred, it becomes a lot more difficult. So the missing H5N1 data from Minnesota cattle outbreaks likely artificially inflates estimates of poultry to cattle transmission in Minnesota. I did just want to also mention that, it's interesting, this pet food outbreak. I mentioned the Oregon cat, but as we get more sequence data from feline outbreaks, we see that frozen pet food outbreak that was sourced from Minnesota caused infections in 3 different states and infected a wide variety of different types of felines. These are not wild. These are in wildlife sanctuaries or zoo animals, or that's how they were identified. Finally, recently the USDA has rolled out this national milk testing strategy to test pooled raw milk on farms and identify subclinical infections. And through this, we were able to recently identify a case of B313 on a dairy in Stearns County and link it back to on the phylogenetic tree to the same farm that experienced an outbreak in July of 2024. So this is the exact same farm 9 months later. On the tree, it looks like this has been persistent infection on this one farm. Obviously, it would be nice to have more data from intervening months, but it raises a lot of questions about H5N1 persistence and how long the virus can persist on a single farm. So in conclusion, Minnesota's B313 outbreak during the spring of 2024 was seeded by multiple independent virus introductions. At least 3 South Dakota cattle are the most likely source. And even in Minnesota, cattle also were likely to be the source of the B313 outbreaks in turkeys and chickens. Minnesota turkeys processed as pet food sourced a multi-state outbreak in felines, and routine surveillance of raw milk is needed to detect long-term persistence on dairies. Thank you.
All right, we are kind of making things up here now, okay? So, which is fine too. Ian suggested we have a few minutes for Q&A, and I imagine you have a question, Ian, or anybody else?
My question for all of you then is, how many of you think that the most pressing issue around H5 in Minnesota is human health? Raise your hands. The most critical thing to address in Minnesota around H5 influenza is human health. How many of you, okay, 1. How many of you think it's cattle health? We didn't hear about cattle, but we know there's been a big outbreak. Nobody? Scott Wells, you need to raise your hand on that one. For God's sake, you're getting paid to say that one. Okay, so food security or feeding the family or human health. So is the major threat to Minnesota related to humans or other things? Scott, did you have a question?
I think your question, Carol, really lays it, really helps us to think about the whole theme of this, right? Which is One Health, which gets to Tiffany's comment, right? All these things fit together. And I can say that dairy cattle are not the most pressing issue, but they're clearly very important. But it doesn't have the same impact at all as it does on commercial poultry operations. And it doesn't have the same impact on, people that have to buy eggs and things like that. So it's really hard to kind of talk about the most important. These are all important, but it's a One Health issue. We have to work together. I guess that's the key point.
Okay. So how many of you think that the solutions are things that you could invent or you could implement? That the solution to H5, to controlling H5, for all of the species and the environments and all of the problems that it's causing for all of us are within your grasp, within your skill set. Collectively, in veterinary medicine, what would you do? And what other disciplines? Because I'm going to tell you, I talked to, I think, some of the people that are solution makers who would be ag engineers, right? Those are people who are going to design new buildings. Those are people who are going to design new strategies for how to separate wildlife and animals without, without damaging either. So how is it? So what do you think they think One Health is? They thought it was an insurance company when I asked. And Alonso talked to CBS and they said, that's going to turn us off because we don't see ourselves in it. So if we can't figure out the mechanisms of basic science that connects us all, or we can't figure out some of those things, maybe it's communication. No? Solutions?
Okay, I'll save you guys. There is a lot of opportunities, right, for us to work on this, and these are very tough questions, and it spans the gamut of levels that we need to be working on, right? The communication aspect and education, both others as well as ourselves. I am still very ignorant about One Health, but I learned a lot just preparing for this. And I think we're probably all on that same kind of trajectory. Some of us are working on this a little bit longer, no more, but a lot of us still not quite. I think the work continues, right? Hin, your question, we almost forgot. Okay, here.
Since we're talking about basic science, I'm going to nerd out with Declan here for a moment. So you said that you found 5 raptors positive, and you sequenced the full-length genome, and it's actually the D1.1 genotype, and it's not the more prevalent B3.13. And we know that D1.1 can infect wild birds and other mammals, including cattle, right? And also the D1.1, if I remember correctly, is actually, maybe Beth can tell me about this too, there's some evidence that maybe D1.1 in humans can cause more severe disease than the B3.13. So what do you think is going on with these raptors? Where do they pick up the D1.1? And are you concerned that it might be zoonotic and transmitting to human? And, is this something that we should keep an eye on from the raptor perspective?
I'm not saying that all the variants, because we're only starting, right? I'm not saying that all the positives we're getting is going to be H5N1 or D1.1. So for those who are familiar with this annotation, so there is a group which is where you can split this virus into 2 main categories. The original introduction was this B313, right? And that circulated first, and then now we start seeing more D1.1, and that's one of the ones that's been linked to some of the dairy industry. But it doesn't mean it's the most prevalent. It doesn't mean it's the only one. There's also others around, and we are only getting what we are seeing here within Minnesota. This is all relatively new. This sort of depth of analysis is something we're trying to get our hands on to understand what true diversity is out there. But even in our data set, it's a really small data set.
So let me back up. You got some false premises here. First, D1.1 is the most prevalent wild bird virus out there right now. B313 was the virus that infected cattle. It was in a few wild animals and birds early on. The single crossover event happened in Texas. And then there were multiple species that came away with that, mostly around poultry farms or around dairy farms. Based on that surveillance, it was never found in wild bird populations. So the D1.1 is the most prevalent. Here in Minnesota, our cases most recently have been mostly D1.1. We now have a D1.3, a 1.5, and a 1.6, which is the H5N2. We also have several that, since the federal government shut down, haven't been typed that are going to turn out to be D types. So the H5 segment that we're talking about here, when it's named D, it's coming from the A3 group of introductions. There were 6 introductions back in 2021, 22, and those 6, B was the type that came from the A1 group, A2 group, A3 group. So D is from A3, and so we know that A3 is what we've been dealing with for a very long time. They are all, A1 through 6 are all 2.3.4.4B. And so we had previously in 2014-15, we had 2.3.4.4 C, and so that's a very, very different type. So the
what's circulating in wild birds right now is D1.1. So that's where the raptors got it.
And just to make another like virology point about what the potential for influenza and looking back at some of our historical pandemics, one of the things that's really challenging is the ability to mix and match segments during co-infections. And some of the outbreaks, human outbreaks that we've seen have resulted in reassortments of even 3 different viruses. And, we often talk about it in terms, historically, we when sequencing was more limited, we would look at, hemagglutinin, which is the surface glycoprotein that mediates entry by interacting with sialic acids or other sugars. So that's not the only virulence determinant. Like we kind of look at that because that was the easiest thing to look at. But there's, depending on whether you're talking about A or C, influenza A or C, you have 7 or 8 segments total, and those do other things. There's polymerases, there's interferon antagonists, and it's potential that there's changes in those other segments that could mediate differences in pathogenicity. And I think that's the argument now, because we have the technology, is we need to look at the whole genomes and really be able to pick up when these reassortments happen, because you could see other effects on pathogenicity that may not be predictable by looking at HA alone.
Wow, fantastic. Sorry, Declan. I know you guys virologists could be talking about viruses all day long, right? Thank you all so much for this part. Thank you, the speakers. Let's go outside, have some refreshments, and continue the conversation. Thank you all.